In this lab, we investigated the separation of a mixture using chromatography. We specifically investigated the two phases that are used during chromatography, stationary phase and a mobile phase. Our hypothesis was the number of colors in food coloring dye can be identified by performing paper chromatography, which was one of our techniques. For our safety precautions, we washed our hands before and after the experiment and also wore goggles throughout the experiment. For disposal, we made sure to wash the test tubes thoroughly and also to dispose of the test tubes properly in a trash bin. For materials, we used 4 pieces of chromatography paper, 4 medium test tubes, 5 toothpicks, 4 FD&C food dyes, specifically red, blue, green, and yellow, a ruler, and a test tube rack.
Hi, Ms. Risi. I'm Andy, lab team member 2, and my job is to explain the experiment. So for the procedure, first we need to wash our hands in case that our hand may contaminate the chromatography paper. Then we dye the spots 3 to 4 times with toothpick on the chromatography paper, as shown on the top right GIF. We dye the spots 3 or 4 times because we want to make sure there's enough dye on that dot and it can travel and reach its maximum distance. Then we put water into the test tube and make sure the spot is above the water. This is because if the spot is below the water, the dye will diffuse in water and it won't go up. We secure the paper at the neck to make sure that the spot is always above the water. Then we wait for about 20 minutes. We wait 20 minutes because we want to make sure the time is enough for the dye to travel. And the growth of each dye is shown on the bottom GIF. And after about 20 minutes, we take out the chromatography paper and measure the distance that solvent and dye travels. And the bottom right GIF shows the moment when we take out the paper. Then we get our resultant data. So the right picture is our result. And based on the result, we found that some dye, and which I mean is green, is composed of 2 different dyes, which is blue and yellow. And by measure the length of the result, I mean the distance for each strip that the solvent and dye traveled, we can get our solvent length and color length. And then we can get our RF value because RF value is equal to color length divided by solvent length. And from the RF value, we notice that each color has a unique and different RF value. And that's pretty much my presentation.
Our team found that the yellow dye was moving the slowest along the paper and it moved the least, while the pink component of the red dye was moving the fastest while moving the most of all the dyes. This suggests that the yellow dye has a high affinity for the stationary phase and a low affinity for the mobile phase, while the pink component of the red dye has a high affinity for the mobile phase and a low affinity for the stationary phase. To reiterate what Andy said, the RF values are found by dividing the distance that the pigments have traveled by the distance that the solvent has traveled, and we found that the further a dye moved, the lower the RF value came out. This can be seen on our chart. For example, the pink dye travels the furthest and its RF value is 2.894, or sorry, 0.2894, while the blue dye, that was a component of the green dye, travels at the least distance and its RF value is 0.9221. As for our errors, we must have added more water to one of the dyes than the others, but all of our measurements seemed correct because we compared with other groups and our RF values were similar. An area that may require further research on our part might be how the polarities of the different dyes fluctuate. This is important because the polarity of the dye is the reason that it will travel a far distance or a short distance. If a dye is a polar molecule, it will prefer bonding with the water and move up the chromatography paper because of capillary action, and if the dye is nonpolar, it will not bond with the water and not travel a far distance.
We completed the calculations and post-op questions together as a group. In order to calculate the RF values, we divided the distance the pigment traveled over the distance the solvent traveled, and we found that the green, red, and yellow dyes we used were mixtures and had several dyes within them. So for the RF value for red, it was 0.8397. For pink, it was 0.2894. And within the green dye for blue, it was 0.9221. For blue-green, it was 0.7357. For yellow, it was 0.6071. For blue, it was 0.8541. For yellow, it was 0.768. And within the yellow dye, orange was 0.64. For 1A of the post-lab questions, the most polar pigment for us was blue because the RF value is the highest, and the most nonpolar pigment is pink with the lowest RF value. For 1B, the water molecules are cohesive to each other because they are polar. Polar molecules are attracted to each other, and since the paper is also polar, the polar water molecules adhere to the paper. For 2A, dye C is the least polar because compared to the other dyes, C travel the most from the origin to the solvent front. Since the solvent is nonpolar and the chromatography paper is polar, the least polar dye will have the greatest attractions to the solvent and travel the farthest. And for 2B, dye A is present in the unknown sample because of the similar ratios of the distance traveled by the solvent front to the distance traveled by the dye in both chromatography papers. Dye A and the unknown sample both traveled approximately half of the distance of the solvent front distance. Overall, we successfully performed paper chromatography for 4 of the food coloring dyes and calculate the RF values of each color found within the dyes to determine the affinity or attraction each color had for the mobile and stationary phases. Thank you for watching.